Thank you very much for inviting me. It's kind of scary. It's a great honor. But what I'm telling you about is some of it is quite old. It's quite a kind of old-fashioned. So some of this, what I'm describing here, comes from the days of VT100 terminals when visualization was done with ASCII characters. And when the very first multiple alignments I made were done using a word processor called WordStar. And that was kind of highfalutin because before WordStar, you did it with line editors or with bits of paper. So it's kind of old, but it's still there and it's still important, and there's still a lot of stuff happening. And so anyway, thank you very much. I said it was a bit scary being here because after some of the talks we've seen, some of the visualizations I'm going to show you were made on the plane on the way over, so they're kind of low-key, so I apologize for that. But anyway, I'm going to tell you about multiple alignment, which is where you line up sequences together. For this talk, I'm basically only going to talk about proteins, amino acid sequences. Okay, this simplifies things. RNA alignment it's a problem in its own right. It's a different world. I'm not going to talk about genomic DNA alignment either, or next generation sequence alignment, which are also big, obvious problems. Let's just look at amino acid sequence alignment, okay? And it arises due to the fact that amino acid sequences evolve, they diverge. When sequences diverge, they accumulate differences, they lose bits or they gain bits, and because of this, it ends up as a computational problem, how to line them up such that the homologous residues are lined up together in columns. And you might guess, if this is a correct alignment, then that the ancestral sequence of all three of these had a W at this position in this alignment. It may not have done so. As a computational problem, you describe it in terms of wanting to maximize the similarity of stuff in the columns and minimize the number of gaps somehow. And that can be done, but it's an NP-complete problem. Traditionally, get back to that later. You need to do multiple alignments because of orthology. That's where species diverge. So if you have one species that diverges into two, then all of the sequences that were in the ancestor, then you end up with two copies of them. And when you end up with all of the species that we have on the planet today, each one of those has its own set of genes, many of which are related to sequences from other species. But you also get something called paralogy, where sequences within a genome can also diverge. And this takes over. Most of the genes in the human genome are highly paralogous. That's probably that's probably not allowed to say highly paralogous, but they've duplicated many times. They belong to big multigene families. One of the most famous of which is the protein kinases. And this is a visualization from a paper in Science 10 years ago by Jerry Manning, who's from Cork in Ireland. That's neither here nor there. But this is a tree of just the human protein kinases, and you can't see anything except these colors correspond to different functional groups of kinases, and they're also evolutionary groups. And these are just the human ones, and the mouse genome will have its tree of protein kinases, and so would all of the fish and all of the birds and so on. So there are an awful lot of protein kinases, and so these arise through paralogy. And I remember there are 500 of them in humans. And this is a multiple alignment, then. This is the first automatic multiple alignment I ever made. And the reason for this is because the globins are the original example. It's not the original, in terms of science, in terms of how we figure things out. The globins are kind of the original example of gene duplication that people knew of. And so they're also the original example of protein structures, because all of these things have a very similar protein structure, as it turns out. These are 7 examples, all of which had their protein structures figured out pretty early on. They're highly divergent from each other. And so it's a very nice original sort of test case. And it's one I keep going back to again and again. As I get older, sometimes it's the only alignment that I'm left with anymore in my world in terms of giving talks. But in terms of visualizing this, there are various things that you might want to do. And so the first thing, obvious thing, is that you'd like to look at the distribution of amino acids in columns or the distributions of the gaps. When we did it first in ASCII terminals or in ASCII printers, you tended to use ASCII characters to summarize some columns. These have shifted. I'm sick of computers. I've been using computers all my life. I spent half an hour of my life trying to figure out why this is shifted by half a character. Okay, and I gave up. Okay, computer won. It beat me. So anyway, these, even though this is Courier, okay, so because I like Courier. I was brought up with fixed space fonts. Anyway, so anyway, they use various ASCII characters to summarize identical columns or very similar columns, and so on and so forth. You can, if you, back in the old days with pencil and paper, you would color in the positions of the alpha helices, which aren't really all the same length, but if you pretend that they are, they come out in these nice, neat blocks. And so these things all have the same, roughly the same 3D structure. You can color in important residues and so on, okay? And of course, these days, there are nice packages for doing this. We've got graphics, machinery and graphics processors and terminals, so you can do much nicer jobs on it than I'm doing here. Another thing that you might want to look at is the relationships between the sequences, okay? And the classic way that biologists like to do this is with a phylogenetic tree or a clustering or something that looks like a tree. Or you can do it with multidimensional scaling. But anyway, here is a tree of these sequences, okay? And so how do you go about doing all of this? Well, here's one way of doing it. So the chairman of this session introduced me, said such nice things about me. I quickly inserted this slide. So this is the JALView package, which the PI, and this is Jeff Barton, and right now Jim is sort of, well, that's you, or it's you. You're the doer. Okay. And so so all of those visualizations I showed you, plus about 1,000 other ways of doing it, it's all in here. And there's a workshop on it tomorrow, so if you want to find out how to do it. Okay, so this is a very good way of doing it. There have also been some nice posters. So this, you know, and there are other visualization packages, just one that I know about, and which works very well, okay? So is that the end of the story? Why not? All you have to do is get my program to do multiple alignment, and then get JALView to visualize it. So, you want a question, okay? So a question from me, a biologist, to the visualizers, the professional visualizers in the audience. You want something to visualize. So here's something, okay? What happens if you have a huge number of sequences? Say 100,000. The reason that we ask this is not just a sort of case where I had to think of a question, I sort of just made this up. Over the last 6 months, we've been making alignments this size. And one weird problem that we've been faced with lately is we can't look at them. And it's kind of disconcerting because the first alignments I ever made, I made by hand. And as I've always been used to, every time you make an alignment, of looking at it, to look for stuff, to see if there's a problem, to see if sequences are back to front or missing or whatever. We've been making these alignments and we can't even look at them, okay? And there are some alignment viewers here. And so there are some solutions. I'll get back to that in a moment. Just give you a couple of examples. I said I wouldn't talk about RNA. But there's a hell of a big RNA alignment of small subunit ribosomal RNAs. There's 1.5 million of them here, and that will grow. Okay, there's going to be more. This is a horrendously complicated alignment in its own right, but let's forget about RNA. In the protein world, there's a database called PFAM, and the biggest family in PFAM, it's the bacterial ABC transporters, has over 100,000 sequences, and there are other families that are that size. Metagenomics is the metagenomics community have huge alignments. They're going to get bigger. Here's another reason why alignments are going to get big. This is the Genome 10K project. Everyone's got to have a big genome project, and so this is a project to sequence every vertebrate genus. Okay, every genus of frog, toad, fish, bird, mammal, everything. It's quite an ambitious and amazing project. And if they do all of this, if there are 500 protein kinases per genome, you are going to end up with 5 million protein kinases and 5 million G-coupled receptors. And that's just those two families, and that's just vertebrates, okay? So there are some very big alignments coming down the line real fast. And so this is an opportunity, and well, it's a problem, but for us, it's an opportunity. I was going to say it's a problem, but the problems are sometimes this community thrives in problems. So just while I'm at this meeting, so Rod Page showed me this morning, and seeing as Tamara is in the room, so this is related. So you knew about this because of the tree juxtaposer, which is a way of scaling, or different levels of scaling of a tree, or in this case, of an alignment. So I just heard about this morning, so I better check. I'd like to check this out. Get the impression this is mainly for long sequences, for genome-sized sequences, rather than many sequences. Although you could use similar techniques both ways. Okay, there are lots of posters just in today's session. So there are these. This poster here as a way of recoding the alignment as a grid of numbers, and that scales to any size alignment. Okay. Here's another one, and this again looked like was good at encoding genome sequences, long sequences. They are also looking at how to do them this way. But anyway, They're just to mention two of the posters, because these are still up today, okay? So in this talk, I want to tell you about a couple of things. So first of all, this isn't visualization, but seeing as we spent the last 20 years of my life doing this, I want to tell you about how to make multiple alignments. There's only 2 slides, okay? But how to make very big ones, and just to try and convince you that you can make them. In order to make very big multiple alignments, you need to cluster sequences. And so clustering is a major visualization sort of angle. So that's of interest in its own right. I just want to tell you a little bit about that. I'm not a clustering expert, but we've been looking at how to cluster a lot of sequences. The next generation sequencing guys and girls have ways of clustering lots of sequences, but they've got a different problem, okay? So they can cluster much more than that, but they cluster in a different way. The kinds of clustering we need are different, and they tend to be harder to do. They're slower, okay? They're more computationally demanding. So I want to tell you a little bit about how to do that. And it turns out the key to be able to make an alignment of 100,000 sequences is to be able to make a clustering of 100,000 sequences. And I'll show you a couple of pictures, some MDS pictures of sequence alignments then, okay? Just to show some visualizations, I suppose. So how do you make a multiple sequence alignment? As an exact, if you formulate the problem as a clean-cut exact problem, we have a scoring function. You try to find the best possible alignment. It's NP-complete. When I first came across this, that globin alignment I showed you, if you had come up with an exactly optimal, exactly best globin alignment, giving a scoring function on a machine that I first edited the globin alignment, that would have taken 2,500 years to compute that little 7-sequence alignment. These days on this laptop would still take probably a few months, okay, for that 7-sequence alignment. So that's a very tough problem. So almost all the alignments are made using heuristics. Probably 99.99, maybe 9999, well, very few alignments are made with anything other than what's called progressive alignment. So it's a greedy heuristic. It was very biologically based, okay? You make a phylogeny first, or a tree, or a clustering, and follow the clustering. You try and follow the evolution. Lots of people sort of simultaneously came up with variations on this. The earliest paper is by Hokeveg and Hesper in the Netherlands. But this kind of disappeared because no one knew how to pronounce Pauline Hokeveg's name. And so, and she was a mathematician rather than a biologist, so that kind of got lost. Okay, the biologists didn't really see that. Russ Doolittle is a famous biologist, so people notice this more, and he coined the name progressive alignment. And Jeff Barton is in there, it's the same Barton, when he was a PhD student, and I'm in there, that's when I was a postdoc. This is the second paper I published as a bioinformaticist. I had older papers on spider distributions in Ireland from a previous existence, but anyway, that's another story. But my second bioinformatics paper was on multiple alignment in 1988. So as to how you do it, I'll just show you a picture. Here's the globin alignment. This is what you want to end up with. You cluster the sequences. You do this by comparing all of the sequences to each other. You get an edit distance between every pair of sequences, and you cluster those distances, okay? And so that's relatively fast to do. It's n squared. And having come up with your clustering, you can then start with the 2 closest sequences in your clustering and align them together. And once you align them together, you fix them together. So you reduce the size of the problem by 1 sequence, okay? So this is the greediness. You can't take those apart again. They get fixed. You pretend that that's then 1 sequence with 2 letters at every position. And you do the same for the next 2. You'll align those together and fix them. The next step in this tree is to align the pre-aligned beta globins with the pre-aligned alpha globins, and so on. And that is a very fast process. I didn't invent this, as I say. I thought I did. I was in the bathroom one day. I thought, geez, I know how to do multiple alignment. My God, this is brilliant. You know, I started coding it up and everything until I realized I'd actually read 2 of those papers that I showed on the list earlier. But I didn't understand them. And there's a lesson, okay? Anyway, so at least I realized it, you know. So we made this program, Clustal, which is still very widely used. This is a later version. This is a graphics version, but in the first version was an ASCII text version. Scientifically, so why so many people use our program? You would get tons of citations. Why so many people use our programs rather than the other programs? It's very prosaic and it's not very scientific. It's nothing to do with scientific wisdom in the conventional sense. It's because we made the program free. There was no restrictions. I begged people to take it off me, even to steal the code if necessary. Just take it. This is before open source. I made it very easy to get. This is in days before the internet. We made the program user-friendly. The program had a manual. The program worked. So that means that you actually test the code carefully on different platforms. And most of all, it worked on PCs, which meant in those days that lab biologists could run multiple alignments. They didn't need to go and see a geek who had a workstation who knew Unix to do a multiple alignment. This is days before the World Wide Web, okay? And so this is all very prosaic, but this is how it happened. So we made lots of versions over the years. It's still going. The last version was version 2.0. That's the last paper we published on it, where we had to recode tons of it into C++ and with the Qt graphics library and so on. But it still looks like this. Recently, we froze it. We tried to freeze it at version 2.1 about 3 months ago. This becomes it becomes difficult to maintain this. We still get like about 10 help requests per day on how to do this. Every time there's a new version of Windows or whatever, you end up having to do a lot of recoding, and it's getting quite tough. So we're trying to freeze it at version 2.1. It's now open source officially. And we're hoping that we don't have to go to version 2.2. Like, we'd like that to be the end of it for now, because there's a limit to how long we can just support this. Anyway, and it's mainly been supported recently by Science Foundation Ireland. So what about the complexity of multiple alignment? When we did this first, complexity wasn't really an issue. It was just very fast, okay, and used very little memory. To make the guide tree, it's polynomial, okay, it's quadratic, mainly. This is a great simplification, okay? Forget about sequence length. Just in terms of number of sequences, crudely speaking, it's quadratic on a number of sequences, kind of trivially. You have to compare every sequence to every other sequence, okay? So that's n-squared comparisons. To do the multiple alignment, once you got a guide tree, that's linear. Roughly, again, to a crude first approximation. Depends how you do it, depends how the sequences are. But roughly speaking, it's linear. So the surprising thing then is that it's the guide tree that stops this scaling to huge multiple alignments. Up until recently, it's kind of tough to do automatic alignments of more than 10,000 sequences. The various packages out there claim they can do it, but you usually have to press all of the buttons on the command line, like minus fast, minus very, very fast, minus no iteration, to really sort of tweak it, and then stand back and wait a long time sometimes. And it's mainly because of this. And that's for two reasons. One is because of the memory needed to store an n-by-n distance matrix. Suddenly this is gone. Over the last year, memory's gotten unbelievably cheap. So that's kind of disappearing. But every one of these n-by-n comparisons is an edit distance, and they're slow, okay? It's an alignment distance, and each one of those gets slow. So when you end up doing 10,000 by 10,000 of those, that takes a long time. And this is what stops the method scaling. There's one method before we came up with our method for making a scale. This comes from 3 years ago from Kazutaka Kato in Kyushu in Japan. It's part of the MAFT package, where I just describe it very simply and very crudely. You take a small subset of the sequences, randomly or intelligently, whatever. I think he does it randomly. You do UPGMA, average linkage cluster analysis, on those. You then cluster the rest in on top of the pre-existing clustering. So once you've done this, this is a little n squared, that's fast, and then this is linear with number of sequences, okay? And that's part of the MAFT package. That gives you fairly crude clusterings, okay? So to make up for that, they iterate. Okay, so they come up with a crude clustering first, they make a multiple alignment from that, and then they repeat it, okay? We were looking, while this paper came out, we were looking at what are called embedding methods, I think. This is where I get scared, talking to computer scientists. I'm probably using the wrong word. But this seems to be, there's a little subset of the clustering literature out there that seems to be called embedding, where what you would like is some coordinate space where your sequences sit, where if you can measure the distance, where each sequence is then located by a vector in this space. And if you can replace each sequence by a vector, you can compare the vectors to each other. Vector-vector arithmetic is very fast. It's trivially fast. Okay? To get the distance between two vectors is an extremely fast piece of arithmetic. Okay, so that's much faster than edit distances. They're very fast to cluster afterwards. So, for example, if you have 10, if you can convert your 10,000 sequences into 10,000 vectors, you can do UPGMA on that in less than a minute just on a laptop. And the process of converting these to vectors, I can talk about that in a minute. They're very shortcuts that you can take there. And if you don't have enough room to store the distance matrix, you can use, for example, k-means analysis. And so k-means can cluster 300,000 sequences in a few minutes, again, on a single core. And so we took this, we started We tried not to reinvent the wheel. We started by doing what we thought was the proper thing. Went to the computer science literature, tried to find some papers on this. This paper here was concerned with clustering bibliographic text, where when you compare two items of bibliographic text, it's similar to comparing sequences, where you've got edit distances, okay? And they tried to come up with a clustering method which would minimize the number of comparisons that you would have to do. And they came up with this, with an embedding method that was n log n. We tried to implement this, and it scaled okay, but it wasn't very accurate. At least it didn't work out very accurate for our purpose. We didn't maybe give it a fair run, but we did redo it. We did try using this. We also tried this method here, sparse map, which had been applied to a protein problem. They said that this would work with enormous protein problems. It's a bit suspicious that the example they gave in the paper was just to cluster 300 sequences, but they said that this could be used for clustering all of SwissProt. We clustered this. The clusterings were very good, very high quality, but it was very slow to run. It didn't scale very well. We ended up in desperation just inventing what is almost a crude subset of those two papers I just showed you. It's just about the simplest possible way that you could think of doing this. So it's kind of trivial, but we went the hard route. We tried the hard ways first before we did this, okay? So again, you select a small subset of the sequences randomly. There are different ways of doing that, kind of randomly. You take log n of the sequences as your subset. You then use the distance of every sequence to each of these k reference sequences as the distances in the coordinates in the vectors, okay? And you use various heuristics because, you know, if two of the reference sequences are identical to each other, well, that's a waste of time. So you'd like to avoid identical sequences, and you'd like to use the outliers. We did this in order to make the paper look more intelligent, make the paper look clever. It turns out, if the data set is big enough, just randomly works fine. Okay, it doesn't matter if you've got duplicates, and it doesn't matter if you've got outliers. If the data set is big enough, if k is big enough, it really doesn't matter. Anyway, ultimately, that gives you an n log n clustering method. Now, the computer science literature is awash with clustering methods. This is going back to the 60s. So this is just a graphic to summarize that. Instead of computing the entire n by n distance matrix of sequence comparisons, you just compute these blue stripes, which gives you n by k distances to compute, which is much faster, and that is then easy to cluster in different ways. As a byproduct of this, you can take those vectors and just do PCA on them. And remember when I said this to the student who did this, I'll give you his name later, show you a picture of him later. We had a nice example. He was looking at influenza sequences. So here's a nice example of influenza sequences. Going from 1967 in this blue here around to 2008 in this orange here. So the colors, please, given what people have been saying, those colors sort of look nice aesthetically. You won't be able to divide the years up very well. This is a 3D PCA plot. I'm sorry, Tamara, forgive me. If I could explain how this happened. It came, okay? It came into my office. He says, oh, I've got this wonderful 3D PCA plotting program. I said, oh no, not 3D PCA, that's terrible. You know, I'm a grumpy old man, don't show me stuff like this. And he said, no, but look, it's beautiful. And it's okay, you know. But it turned, I wanted him to do a normal, just a 2D plot, but it turns out he spent ages coloring these, okay? And I didn't have the heart to make him do it again. So this is the only picture we have of that data set, okay? So just pretend it's 2D. So that's the visualization. So that part is okay. What's more important is, are the clusterings any good? And measuring cluster quality is kind of hard, okay? It's complicated. There are many ways of doing it. There's a huge literature on how to measure cluster quality, obviously. For our purposes, we're lucky. We're making multiple alignments. You can benchmark multiple alignments, okay? That's not perfect, it's not ideal, but you can do it. Here is one benchmark example alignment. This is a PFAM family where we have a reference alignment that we believe to be true. The sequences are relatively distant, only 22% identical. And along the x-axis here, we show the score varying from perfect match to the benchmark to no match at all. When you run Clustal, it's a complicated figure, sorry, but anyway. The pink arrow shows the score that you get from a default Clustal alignment, so it gets about 65% of the positions correct. This is a histogram showing 1,000 random trees. You take the correct clustering, swap the labels randomly with each other. And so that shows you why you use a guide tree. You see, that's kind of cool. that even the very best of the random clusterings is still only, Clustal one is still better. So that's why you need a good clustering, okay? So it really does matter. These are then 1,000 sparse map trees. I mentioned sparse map earlier. The trees from sparse map are good, but it doesn't scale so well. It is n log n, but it's a bad n log n, at least the way we coded it. Maybe we didn't do it right, but we found that tough to run. And here's embed, our method. So it's a tiny loss in accuracy compared to the default Clustal, but with a colossal speed up, okay? And so when you do this, we are then trying to make a new Clustal program called Clustal Omega. This is supposed to be me hinting that this will be my last ever Clustal. So if you excuse the biblical reference, it's a bit pompous, I suppose, but anyway, we have T-shirts. So, sorry. If the program is a success, the T-shirts will be readily available. We'll have a merchandising site, and we'll move on to other items of clothing and cookware and so on. If the program is a disaster, this is the last you'll see of all of that. Anyway, we're hoping to release it soon. We sent out a beta version, and we got some bugs back, but we're hoping to release it soon anyway. It scales really well. It uses Embed. So it can make huge alignments. We've replaced the alignment engine with a HMM alignment engine. A thing called HH-Align makes really good HMM alignments from Johannes Söding in Munich. We do something that arose from a conversation with Kevin Karples. And Kevin Karples is an old friend. I know a lot of people here know him. I tried to find the least geeky, least hairy picture I could find of Kevin. And so this is him wearing a nice blue sweater, which is unusual for Kevin, which is possibly on the right way around. I'm not totally sure. But anyway, so from a conversation with Kevin, we figured out a way of trying to reuse an alignment. You make an alignment and reuse it later to help you align new sequences. So anyway, we have a logo. It says also something about graphics. So a guy in the lab made this logo. I said, ooh, cool, like that, okay? This guy, Dave, he's a software engineer, but he worked as a graphic designer for a while, okay? And he looked at the logo and said, oh, horrible. Okay, so there's something about the communities where professional graphics designers know that that can be done better. And I have no idea why, except when they do it, it looks better. Okay, so this was done by a bioinformaticist. So anyway, it's a first approximation. These are the guys who are doing the coding. So just as an example, so you can then align 120,000 relatively short sequences in 6 hours on one core. And using OpenMP, it scales fairly well. So you can get huge alignments, and it scales up to alignments of any size, we're guessing. But this is yeah, anyway. Okay, and it's a fairly accurate program, and we're hoping it'll come out soon. So, the last bit of this talk, yeah. So MDS PCA. I realize there's a bunch of people in this room know more about this than I will ever know. So I'm kind of an outsider to this. I treat this very much as a biologist. But anyway, one way to visualize stuff in alignments is, of course, to use dimension reduction techniques. And so one way of doing it is you can do principal components analysis if you treat the alignment columns as variables, okay? Or something that we like is correspondence analysis. It's a technique invented by the French statistician Jean-Paul Benzécry, which is very nice for alignments. And we can talk about MDS in a moment, get back to that later. Here's a JALView picture. This is just to show you an alignment. JALView has PCA in there somewhere, or PQORD, MDS, one of them, okay? So to do PCA in this, what you have to do is take every alignment column and turn it into 20 binary variables. So it's really clunky. This is, evolutionarily speaking, pretty grim. It's an incredibly crude model from various ways of looking at it. It allows you to do the PCA or the correspondence analysis. You can also encode the amino acids with physicochemical properties, and that also gets away from evolution. That's nice in some ways, but anyway, it's pretty clunky, but it scales really well. You can do PCA plots of any size. Very easily. Here's one example I want to show you. So this is an example from Rob Russell. We're looking at 3 groups of proteins that are very similar to each other. We're asking which amino acid positions determine the specificity of the groups. So it's kind of a supervised problem. They were specifying the groups in advance, okay? So we use correspondence analysis. We supervise it with something called between groups analysis, which is a technique from these French guys, Sylvain Dolidec and Daniel Chassel. This is equivalent to using partial least squares discriminant analysis with PCA. Okay, it's kind of a discriminant function analysis. Anyway, here are the plots that you get. In the top panel, you see the proteins. Each one of these is one sequence. In the bottom panel, you've got those shrunk down and surrounded by the amino acid positions, plotted in the same plot. Each dot is one amino acid position. And the these are labeled. It's a pretty grim visualization, but it kind of shows you what I want just to tell this story anyway. Each one of these boxes is then the most extreme ones labeled. So this says that there should be an alanine at position 7 in the alignment, or this says there should be a tryptophan at position 98 in the alignment. These are the 20 binary, every column turned into 20 binary variables. So if you then ask, why are the trypsins different from the chymotrypsins? This is correspondence analysis, and you read those by asking what is the variable furthest out from the origin in the same direction, which is to have an aspartate at position 229, okay? So it gives you these very nice plots for visualizing the interrelationships between the variables and the sequences. And this is a plot from their picture, which is a visualization using AlScript, which shows that position highlighted there, where these are the chymotrypsins, which all have a D at that position and not in the other sequences. So it allows you to do very nice plots. But the encoding is crappy. Another way to do this is you could use real evolutionary distances. Compare your sequences using, from a computer science point of view, proper edit distances, or from an evolutionary point of view, using evolutionary distances. You make your distance matrix, and you try to find a low-dimensional projection that fits the distance matrix according to minimizing stress. If you use Euclidean distances, and that's called classical scaling, and it's equivalent to another technique called principal coordinates analysis, which is sort of independently invented. So I heard about this before I heard about this. My only ever scientific publication on my own was this, where we applied principal coordinates analysis to what I called a large sequence data set in 1992, which was 655S ribosomal RNAs, which was large at the time. How do you do this on a large scale? There are lots of ways of doing this. People in this room know lots of ways of doing these large-scale MDS projections. I just want to show you some that we've tried, because these are just a couple that I happen to know about, and that we tried in the lab and at work. Wenxiong Li, sometimes in Chicago, sometimes in Taiwan. This is his Taiwan group, or some guys work for him in Taiwan. They have a thing called SC, MDS, where you do small MDSs on subsets of the sequences and overlap them. But it's very sensitive to the distances being Euclidean, okay? Because in order to do the overlapping, the distances have to behave well. I showed you Embed. Okay, so Embed was a PCA rather than MDS, but the PCA was based on distances, on evolutionary distances, so that kind of fits here. That scales really well. You can do MDS on a subset of the sequences and just plot the rest in later. Okay, that scales again. Once you've done your subset, you can add in any number, and that's linear with the number of added sequences. One that we really like, that works great, is something called landmark MDS, which I'll show you a picture for this. So anyway, you can easily do MDS on huge numbers of sequences. This is a terrible visualization because it tells you nothing, okay? But if I explain again why we did this, this is just to see if you could do it, if landmark MDS would compute, if you could just do it, just to test out some code. And so we just took a huge wodge of sequences. The alignment is probably a bit silly, a bit meaningless. But this is an MDS projection of 300,000 sequences. It's meaningless because there's no labels. You don't know what any of this is. This is just to see how long it took. So I got this on Monday. I flew here on Tuesday. I got this on Monday morning off Dave Deneen, one of the photographs you saw earlier. I said to him, so how long did this take? He said, overnight. I said, well, yeah, well, sort of, how long? He said, well, I don't know. I didn't time it. It's just, I came in the morning, it was done. So the unit of computation to do 300,000, to do an alignment of 300,000 sequences is 1 overnight. I said, how long did the MDS take to do? He said, oh, it was pretty fast. I said, well, like how fast? He says, oh, it's pretty fast. So in total, to do the alignment and the projection, it takes 1 overnight and 1 pretty fast. So, but I'll just finish with some pictures of something that are a bit better labeled, something where you can actually see a bit of biology. It's a much smaller case. This is Weifeng Shi, who made the picture, the 3-dimensional PCA I showed you earlier. He's interested in flu, so we're doing flu alignments. He took 8,000 H3N2 flu viruses. This is one of the main forms of seasonal influenza. There are sequences in freezers going back to the 60s and beyond. He used proper evolutionary distances, okay? The Kimura 2-parameter distances. These are DNA alignments. You do an MDS projection of these. These are colored according to this. So we color them according to which continent they come from, but ignore this. This is just to show you the colors do mean something. For this talk, you can ignore that. That's just going to confuse you, all right? And these are then I've blotted out all of the sequences except the ones from the 1960s. And if you then show the ones from the 1960s and 70s, they go like this. You get more sequences in different parts of the world. 1980s, sequencing is taking off and freezers were getting bigger and people were collecting more flu samples. In the 1990s, that happens on a much bigger scale. There are systematic programs to start collecting these sequences. In the year, then just 1 year at a time, year 2000, 2001, 2, and so on. So that's just a pretty picture to finish with, okay? You can do this with a phylogenetic tree, and people regularly do. It's just doing an MDS gives you a different way of looking at it. It gives you pretty pictures. Okay, and that's it. I think, sorry, but I've gone a bit over time.